International astronauts and cosmonauts, all members of the Association of Space Explorers, gathered in September 2008 at the University of Washington in Seattle for two panel discussions: International Space Programs Review and Human Health for Long-Duration Exploration. This program is part of a series featuring Dr. Millie Hughes-Fulford, former NASA astronaut, discussing human health for long-duration exploration.
Good morning. My name is Phyllis Wise, and I'm the provost here at the University of Washington. And I want to really welcome you here to the 21st Planetary Congress. We are thrilled to have you here, excited about the work that you do, and really want to have the connections between the University of Washington and you even better. Your work has led to such a better understanding of space, of the atmosphere of our environment, and have developed so many international collaborations that have helped this work go forward. The UW is deeply connected with space. As the home of Boeing, we have been very, very interested in the development of flight and the technology that goes behind that. There are at least 8 different UW graduates who have been involved with NASA space flights, the latest of which is Greg Johnson, who will be piloting the last shuttle flight to the Hubble Space Telescope, and that is scheduled to take off in October, on October 8th. So many of our children and young people, some of whom are sitting here in the audience, dream of becoming astronauts, and we hope that the training and the exposure to the work that you do will encourage them even further. Our departments of astronautics and aeronautics, astronomy, atmospheric sciences, and earth and space sciences are proud contributors to the work that you do. And so I also want to recognize Bonnie Dunbar, the chair of the Washington State ASE Coordinating Committee, who has made sure that these days here that you will spend here are good and productive for you. I just want to say that I know that some of the participants are stuck in Houston, but I'm sure that this Congress will be very, very rich and very important to your continuing collaborations. And what I'd like to do now is introduce the leader, the panel leader of this discussion for you today.
Two of our speakers, Dr. John Charles from the NASA Johnson Space Center and Dr. Jeff Sutton from Baylor College of Medicine and the National Space Biomedical Research Institute, were in Houston safely, we hope, during the hurricane, but unable to get out and to attend today. So we will talk a little bit at the end of the session about some of their work and what they were going to cover. So we have flip-flopped the order of the program today, and we'll start with Dr. Millie Hughes-Fulford. Millie entered college at the age of 16 and earned her Bachelor of Science degree in Chemistry and Biology from Tarleton State University in 1968. She began her graduate work studying plasma chemistry at Texas Women's University as a National Science Foundation Graduate Fellow from 1968 to 1971. She was an American Association of University Women Fellow from 1971 to 1972. Upon completing her doctorate degree at TWU in 1972, Dr. Hughes-Fulford joined the faculty of Southwestern Medical School, University of Texas at Dallas, as a postdoctoral fellow with Marvin D. Sipperstein, where her research focused on regulation of cholesterol metabolism. She has authored and contributed to over 120 papers and abstracts on bone and cancer growth regulation. Since then, she was named the Federal Employee of the Year for the Western Region in 1985, International Zontian in 1992, and Marin County Woman of the Year in 1994. She was a major in the U.S. Army Reserve Medical Corps until 1995. Selected as a payload specialist by NASA in January 1983, Dr. Hughes-Fulford flew in June 1991 aboard STS-40 Spacelab Life Sciences, or as we call it, the SLS-1 mission, the first Spacelab mission dedicated to biomedical studies. The SLS-1 mission flew 3.2 million miles and 146 orbits, and its crew completed over 18 experiments during a 9-day period, bringing back more medical data than any previous NASA flight. The duration was 218 hours. She is a professor at the University of California Medical Center at San Francisco, where she continues her research. As the director of the Laboratory for Cell Growth and Differentiation and scientific advisor to the Undersecretary of Veterans Affairs, she studies the control of human prostate cancer growth with VA grants and the regulation of bone and lymphocyte activation with NASA grants. She was a principal investigator on a series of Spacelab bioreactor experiments which examined the regulation of the osteoblast, and of course you remember all this, the osteoblast bone cell growth. These experiments flew on STS-76 in March 1996, STS-81 in January 1997, and STS-84 in May 1997. Today, she remains a professor at the University of California in San Francisco and is the PI of 4 grants, 2 in immunology with NASA and 2 with the Department of Defense and the Department of Veteran Affairs on stem cell research. Please help me welcome Millie to the stage.
Bonnie, thank you for such a nice introduction. It's my pleasure to be here today and to see many people I know and many that I do not know, and hello to everyone on the webcast. I want to talk today about the human health in long duration. In addition to radiation, we also have problems with bone loss and with immunosuppression in spaceflight. On my mission, it was the first dedicated medical mission that NASA flew. Our people on board was Brian O'Connor, our commander, Tammy Jernigan, who I think is in the audience today, Sid Gutierrez, Jim Bajan, Ray Seddon, myself, and Drew Gaffney. Going into space is not just a 2-month exercise. We were down there training for over 8 years. That training included water safety. We went through a series of trainings at Johnson Space Center in the wet facility where we were taken to the very height of the dome and then dropped into the water. And right after that, we went to Homestead Air Force Base, where we underwent water training with the right stuff. Now, I walked into Homestead. I was the only woman in my class. I looked around, and I was the oldest person in the class. Rest of them looked a little bit like Tom Cruise with the crew cuts and the aviator glasses, and they kind of looked at me and said, "Where did this woman come from?" The next 3 days, we were dragged behind boats, pushed into the water, parachuted into the water. The last day, walking back to the class, I was stopped by 3 of the other students. They said, "Ma'am, we wanted you to know, when we were out there and thought that we couldn't do it, we'd look at you and say, if she can do it, we can do it." This is our flight.
T-minus 15 seconds.
I'm right there.
T-minus 10, 9. We have a go for engine start. 6, 4, 3, 2, 1. Ignition and liftoff of Columbia on the first dedicated medical research flight.
One of the first pictures ever taken of the external fuel tank was taken by Jim Bajan, and you can see the charred area of the top of the tank. The reason I kept this picture in the talk is because of this is one of the few pictures that really shows the radiance of the Earth that all of the astronauts see when they look down on the Earth. And it makes the Earth look very fragile. My first sight of the Earth, I looked at it and I felt like the Earth was alive. And then the scientist, half of me, said, "That's silly," and went back to the scientific part, well, why do you think that? And it's because when I'm in the laboratory looking at living cells through the microscope, it's the same quality of reflection that I get from a living cell, except it was the entire Earth looking that way. This is a common day in a space lab. One thing you'll notice is there's a lot of hard wiring of all of the readouts on the biomedical readouts, and I know that we heard at lunch today that that might be a thing of the past, and we have a potential for eventually not being tethered down by all of the wires. This is my work area where I collected blood. This is just a space lab, and you can see that the trip home to the bathroom and the galley was maybe 8 seconds. Every day, our commander and pilot would fix one meal for us so we could all catch up on what was going on and how things were going for that day. Well, some people say, how do you eat in space? And the answer is very, very carefully, because even scrambled eggs will end up all over the place, and you have to catch them if they get away from you. Oh, this is a fun one. We did have some dry shampoo, and it worked really great on the guys with short hair or no hair, but didn't really work for the women with long hair like Tammy. We just didn't go up there to have a good time, to look out the window. We went up there to study several major systems, one of which was bone loss. Bone loss was first seen on the Apollo missions, where the astronauts lost around 2% of their bone in less than 2 weeks. Looking at data from Apollo, there's about a 2% loss. The first Skylab was fairly short, and there was actually a positive gain of bone on Skylab, but again, in Skylab there was a lot of resistance exercise and putting everything together. Skylab 3, it was mixed. One person lost a great deal of bone, over 7%. And if you look at who lost bone or who gained bone, mainly it was the commander who did not lose bone, but he had first dibs at the exercise equipment. The pilot was second, and the person that lost most bone was the scientist, who was at the bottom of the list for exercise. And on Skylab, we saw an even greater variation of bone loss. And again, it depended upon the type of exercise they were doing. One thing that's been noticed about bone loss, at least in animals, since we haven't taken human biopsies yet, is that there is an arrest line in bone. This was taken from a rat. The animal, at the beginning of the flight, was given a marker that would glow, and then when the animal returned, another marker. Well, in the intervening time, there wasn't a lot of bone growth at all, and there was actually an arrest line that reminds one of a ring on a tree, and it showed that the animal was not growing new bone during the space flight of 8 days. Going back over data for men and women, this was taken from the American Association for Bone and Mineral Research. You can see men and women lose bone at a different rate. Women have an accelerated bone loss after menopause. And the main difference, though, if you look at it beyond the menopause, is the fact that men start off with more bone than women to begin with. The rate of loss during menopause is around 5 to 10% in women, and that is the same amount of bone loss many times are seen in astronauts, even on station where several of the astronauts were on a very aggressive exercise protocol. They didn't lose overall bone, but they were still losing around 5% of the hip. The nicest study to date is a study by Vico et al. That was published in Lancet in 2000. And looking at the tibia, you see we have a group of people that did not lose a lot of bone, and it actually was gained after they returned for 6 months. So these people were on Muir for 6 months, and then they looked at the gain of bone. Then you have a group of people that lost bone, and the bone did not recover with time. And one individual in 6 months lost 24% of the bone. That's 4% per month. And only a very little of it returned. So the long-term bone loss is a problem. We need to look at it and find out what causes it. The data here suggests that maybe we have a genetic feature of bone loss. Maybe some people are more prone to bone loss than others. What do we do about it for long-term missions? We could have an engineering solution. We could build an artificial gravity centrifuge on ISS, Moon Base, and Mars vehicle. And on ISS, we could determine how much time we have to be at 1 gravity or 2 gravities not to lose bone at all. We did have the centrifuge set for ISS, and it was cut. It'd be nice to have it uncut and put back on station. The other solution we could have is a medical solution. We could start genotyping people and find out the genotype that's most resistant to bone loss in space, and then use that as part of the selection for the crew. The other thing is we can find treatments that would increase the needed bone factors that are required for new bone growth. The immune system was also blunted in returning Apollo astronauts. Apollo 7, all 3 crew members had a cold. Apollo 13, one of the crew members became very ill with a very common bacteria that normally does not affect people and usually is only a concern if you're immunosuppressed. And all 15 of the 29 Apollo astronauts had an infection either during the flight or within 1 week after flight. There are 2 types of immune cells that we think are causing the change. One is the T cell, the other is the monocyte. This is the recovery of the first moon mission.
Now the 3 men wearing biological isolation garments enter the mobile quarantine facility and their earthly confinement.
Soon we were to find out that indeed there were no nasty, gnarly bugs on the moon. And the next thing we see is that the measures were reversed. The astronaut returning from space were now greeted by an Earth crew that had an infection mask. This was seen on the return of Skylab. And you'll notice all the people helping him out have on infection mask. This is the same type of thing you see in returning astronauts in Russia. It's the same quarantine health stabilization that we have pre-flight for all of our flights, because we want to protect people from infection, not protect the Earth from some exotic space bug. Now, what's causing the change in the immune system? This is data taken from the Skylab, and we have 6 individuals. 3 are the backup crew, and 3 are the flight crew. And we stimulate the T cells to grow, and indeed they all grow. In the backup crew that did not leave the Earth, we still have people with an active immune system. However, the returning crew, the T cells are not stimulated. They're not growing. So there is a problem in how the T cells are being put together while you're in space. And it took up to 2 weeks for this to recover and the returning astronauts. Now, there's 3 things you need for a T cell to be activated, same 3 things that you need to keep from having an HIV infection. In the real world, you have an antigen-presenting cell. In the lab, we can either use anti-CD3 or concanavalin A to reproduce that. In the real world, you have an accessory cell. That's the second step. In the laboratory, we can use a compound called anti-CD28. The third and final step for full activation of the T cell to marshal your immune system is the production of IL-2. IL-2 is produced after the first 2 signals, and it's produced inside of the T cell. Once IL-2 connects to the 2 constitutive subunits of IL-2 receptor, it then activates the production of IL-2R alpha, which is giving you the full immune system ability. Now, the IL-2R alpha is also one of the molecules that is downregulated in HIV. Unless you have all 3 events occurring, your immune system is not normal. On Spacelab, we see Owen Garriott here, we found out that indeed cells are sensitive to gravity. There are 2 types of cells that are sensitive to gravity. One is the immune cell, the other is the bone cell. Not surprising when you think that we developed in a 1 gravity field that some of our systems might actually depend upon gravity. On SLS-40, we also took blood, we isolated it, put it into a centrifuge, and the centrifuge was a modified record player. And so we had the temperature the same, and then we put the cells at different areas of the record player. So we had a 1G, we had a 0 gravity. And indeed, on SLS-1, we found that in 1 gravity, IL-1, IL-2, IL-2R alpha were all the same. In space flight, in a 1 gravity centrifuge, all the same. However, in microgravity, we found that IL-2R alpha is not made. And so we now know for sure that that's one of the steps leading to the inactivation of the immune cell. Back on Earth, we're able to reproduce some of the gravity studies or microgravity studies using a random positioning machine. Then using a gene chip, looking at all 30,000 genes of the human body. 2 million dots here. And this one, 2 million dots, and the new one's a lot more than that. We found that there are genes that are activated during T cell activation that are turned on that are not turned on unless you have a gravity vector. And so now we're getting very close to understanding why the T cell is not reacting properly, and actually we're going to be able to find the pathways responsible. So a little cartoon, you have your 2 signals, making the IL-2. The IL-2 connects to the IL-2R, making the 3rd one in normal gravity. In microgravity, that's missing, and that's why we have a problem. We're going to continue this work in spring of 2009 on ISS on the experiment called PADIAC. We just finished coming back from Baikonur, October 2007, where we studied the monocyte, which is the 2nd immune cell that's affected by gravity. This is coming off the Energia plane going into Baikonur. Now, I thought we'd be getting the big bus, the scientist, but indeed, we got the little bus right next to, and those were our 2 keepers that were with us the entire time. If you wanted to go for a walk, they were with us in Baikonur. We were on the main military base and didn't have a lot of freedoms, but had a good time. And you'll notice this little bus has all the windows covered. So we weren't supposed to be looking out about where we're going. but that changed on the second trip. Anyway, so the next part of this talk about the immune system I'm calling The Road to Kazakhstan. And this is actually a picture taken by my videographer with the nice shadow of that little bus that we were put in to go over the base. Now, a lot of you don't know what a monocyte is, and maybe you don't care, but it's working in your body right now, so you should care a little bit about it. The monocyte is a white blood cell. It circulates in the blood for about 8 to 24 hours. It can eat bacteria, but not at a very high rate. With time, the monocyte turns into a macrophage. Macrophage means big eater. The macrophage can eat 200 bacteria during its lifetime. Now, the macrophage will migrate to and reside in your tissues where they can engulf invading bacteria, and they can live for several months waiting for an injury to your body. The macrophage, in order to stay in the tissue, has to make adhesion molecules and receptors in order to stay where it is, and also it needs to have strong phagocytic ability. Think of the Pac-Man game that you used to play. That's the macrophage. It's eating a lot. Okay. Now, there are some clues about the monocyte in spaceflight. One is that early on in the 1990s, we saw in rats that had been in microgravity, they had reduced T cells and macrophages in flight. More recently, in 2005, on returning astronauts, we showed that, not we, it was Dwayne Pearson and his group, showed that the monocytes had reduced ability to engulf E. coli. And finally, in a second study, studied in 2000, in 27 returning astronauts, there was a decrease in the percentage of monocytes that had the CD14, CD16 signature that tells you that it's making itself into a macrophage. And so the data is quite convincing. And this is just showing the bars. This is launch plus 10 days. The day of return, R plus 3. So you're actually going to be more susceptible on day 3 than you even are on the day of return. Some people say, well, that's just because you have a lot of stress hormone. Not true. There were no significant changes in cortisol, epinephrine, or norepinephrine. So it's not a whole body effect of stress. It is an effect of gravity. So when we went to Baikonur, we had a hypothesis. We said that gravity is a necessary component for a monocyte to mature into a macrophage. And then by using the analysis with gene arrays of the gene expression in the new 937 monocytes, we were able to determine which genes were involved in the lack of differentiation. Why on ISS? Well, after loss of Columbia, which was our only science shuttle, ISS is the place to do the experiments, and it's a national laboratory that should be made available for everyone to use to do microgravity studies. Now, because of Homeland Security, ESA would not send the hardware to San Francisco. They were afraid that it would be caught up in customs somewhere. So we flew to Amsterdam, and we didn't go there just to look at the windmills. We actually went to Noordwijk, to ESTEC, to start the experiment. You've heard of lab in the box. This is a lab in a suitcase. We took everything we needed for 12 days in that suitcase. People opened their labs to us and made it possible for us to do the experiment. These are the monocytes that we took care of the entire trip throughout the time from San Francisco to Baikonur. Now you might ask me, well, did you have a big incubator? Well, ESA had an incubator that was like that, but we had 11 suitcases, and I didn't want an incubator like that. So we took baggies. We put, you know the heat strips people put on their back for pain? We took those heat strips, we put on the outside the baggie and used it as an improvised incubator for all the trips throughout the move. Okay, then we went from Amsterdam to Moscow. We left Amsterdam Schiphol Airport early in the morning before the sun came up. A lot of people think of Moscow and they think of St. Basil's and Red Square. My people think of Moscow and think about customs. We were held in customs for over 8 hours, and all the time we were worried about ourselves surviving the wait. We finally left the Moscow airport at midnight. We didn't get to the IBMP labs until around 1 o'clock. Got to bed that night around 3. From Moscow, we then went to Baikonur. Lots of nothing between Moscow and Baikonur. The reason that we had to pack very carefully is because there was no place to buy cover slips in Baikonur. One group had lost oil from a vacuum pump, and they were having trouble finding oil to be put into the vacuum pump. So Fisher Scientific doesn't FedEx to Baikonur, so what you had with you is what you got to use. This is in the labs in Baikonur. The hardware put apart. Here's part of the assembly. Took about 18 hours to put it all together. While we were there, we were lucky enough to meet some of our Malaysian colleagues that were doing science in the lab right down the hallway. And we were invited in, so we got to watch some of their procedures while they were working. They're using hardware from BioServe. We were using hardware from ESA. And you might notice that the PI is keeping a close watch on the tech. Thank goodness over here I don't have to stand at attention and watch my people work. This is a picture from the astronauts, cosmonauts going up on that mission. You'll see Sheikh, one of our newest members of ASE, giving a thumbs up. The walkout. We got to watch the rocket taken to the launch pad. And of course, we got a really good viewing of the rocket launch. A little bit closer than you are when we watch a shuttle launch. It was right at dusk. You can actually see it turning dark as the launch. They ended up on station where Peggy Whitson, our first female commander, was in charge. Yuri was the person that took care of our experiment, PKINASE. This is the inside of the incubator. Half of the incubator is taken up by the 1G centrifuge, and then we have the microgravity samples on the side. So the samples are having the same amount of vibration and so forth, and we have an in-flight 1G control. So far, we found out several things. We found that the monocyte lost the ability to mature and adhere. It's hard to see on the screen, but you can see that very few cells are adhering in the microgravity samples, whereas the samples that were in the 1G flight centrifuge matured normally and adhered normally as we expect them to. We are seeing major changes in 1G and micro-G at 3 hours and 24 hours. We have found about 80 gravity-sensitive genes. And using the gene array analysis, we are looking at the signals that cause the changes in gene expression. What are the possible solutions to cure immunosuppression in long-term missions? Again, you have an engineering solution of putting artificial gravity either on ISS to determine how much time you need at 1 gravity to keep this from happening. On moon base, medical solutions. We need to find treatments that improve T cell and monocyte activation and bring it back to normal. I'd like to thank NASA for support of this work and my 2 people, Chai-Fei Lee and Tammy Chang, for all the hard work that made this possible. Thank you.
Do we have any questions?
I work with cancer doctors, and I guess I have a belief that if someone's exposed to a lot of radiation, they may be at high risk for cancer. So in long-term space goals, will we expect our astronauts will have higher cancer risk due to exposure to radiation in the atmosphere?
We have ongoing experiments within NASA looking at the danger of the radiation exposure. Some of the solutions is to shield your vehicle, shield the habitats that you have to keep the radiation exposure down. If we had a new method of propulsion that could get us, say, to Mars in 29 days instead of 6 months, that would go a great way to reducing the radiation exposure. So sometimes you have an engineering solution, and I think that's going to be the best solution for us, because I'm not sure how we can negate the radiation damage with medications at this point of knowledge that we have.
Thank you, ma'am.
Millie, I have a question, and maybe a couple of questions, but let me start out by saying, with all the improvements that we've had, of course, the Russians have had a lot of experience with long space flight, but over the years and now with the ISS, I'm certain we have improved our resistive exercise protocol. Is that correct?
That is correct. I think on station we've had several people doing resistance exercise and actually negating much of the bone loss. But even those guys lost 5% of the hip.
Okay. So as we have improved that exercise protocol, we have made significant reductions in bone density loss.
That is true.
Okay, and do you believe that with continued protocol improvements and understanding, we can get that down to 1-2%?
I think the major thing we need to do now is to understand the genetics of bone loss. And this, of course, would help those of us on Earth that have osteoporosis also, because the number of people that have been on the resistance exercise is still a low number of people. We don't have the genotyping of those people to determine what their genetic background is versus the exercise protocol. So we need more data, but yes, I think that we can reach a point that bone loss will not be a problem if we can get all the data to look at.
Okay, and I know we've had a number of women now involved. Is there, and maybe the sample size I think is still small, but is there a difference in that bone density loss doing the same protocol between men and women?
To my knowledge, there's not a difference. I think, again, it goes back to genetics more than to sex.
Okay.
I do know some data, but I can't tell you.
Sure, certainly. Okay, well, thank you.
Thank you very much. So anyone that's flown, you might want to go get a DEXA scan on your bones just when you get home. It won't hurt.
Millie, I have a question. I noticed you showed that even the Apollo astronauts who did walks on the moon experienced bone loss. One of the things that I think people were hoping for was that, for extended stays on the moon, that doing extravehicular activity would sort of help, would take the place of exercise and help to maintain bone health. And so that kind of data tends to, work against that,
Hypothesis.
that hypothesis, yes. And so, what do you think about? Is there a way that you could make an EVA suit such that it could provide more, better stimulus for bone growth, or a reduction in the amount of bone loss, and what your thoughts are on that?
I think on Moon, even though you don't have full gravity, you have some gravity. I think the resistance exercise that they're doing on station now, combined with moon gravity, would give us a lot of solution to the problem. I would like to restate that I still think we need to do genotyping, and that would go a long way to understanding bone loss, not only in astronauts, but also here on Earth. And I think it would be a huge boon to the entire world.
How entire genotype sensitive for the gravity? Is any change, mutation, or sensitivity to, beside monocytes genes and bone genes, or entire genotype also sensitive for the change in gravities?
Yes, I think in bone we already know several genes that protect people from bone loss, like the Wnt pathway genes, tend to result in people having heavier bone. In immunology, we haven't even begun to look at the differences in who becomes ill, who does not become ill. For every gene that we have the potential for different mutations being present in the body. And so until we know the gene mutations present in the human body as far as some of the pathways for protecting ourselves from infection or for bone, we cannot make projections for some time, except in the bone we do know that the Wnt pathway is a very big player. And I would guess that the people that didn't lose any, yeah.
Can I ask you another question?
Yes.
Is the cycle of the cells every phase changes, like G1 phase or G0 phase? It's became changes in this?
In isolated cells, the work that we did on STS 76, 81, and 84, we found changes in cell cycle of bone cells, definitely. That has also been seen in animal studies, where the pre-osteoblast is kind of stuck during, that arrest line I showed. So yes, there is some information that would suggest that cell cycle, at least in bone, is affected by gravity.
P53 also changes in G1 phase particularly?
I haven't seen changes in G53. We've actually looked at P21, and there were changes there. A P53 and a P21 are two genes involved in cell cycle. Another one is CDK2. Yeah. And we have seen some changes in cell cycle of the isolated cell. Yeah.
Well, thank you very much.
You're more than welcome. What we really need is a centrifuge where we can actually put them on and just measure how much gravity do we need. We know on 1 gravity for the immune cells that we can totally keep the problem from occurring, but we don't know if we need a 0.5 or a 0.6. We don't know the absolute level of gravity that would reverse the problem. And I think we need to know that pretty soon.
Could I ask a question in that vein?
Yes.
Have you taken any data or explored anything about the immune suppression between 0G and 1G?
The data that we have on isolated cells, it's fairly clear that we're seeing changes. Due to the lack of funding for the biological sciences right now, write your congressman if you want to know more, because we need the funding.
All right. Thank you.
Thank you.
I have a question. Thank you for your presentation. You mentioned that ISS is accessible to everyone. Are there some priorities of projects at the moment?
The only reason I'm flying my experiments right now is because I had a liking for the European Space Agency hardware. and I like their onboard 1G control. So I elected to fly with the Europeans, and since I flew with the Europeans, it wasn't demanifested by the U.S. government, so that's the only reason I've been flying these experiments.
Millie, I'm kind of a pilot person, so I'm not real smart. I'm going to say...
Don't say that. I'm married to a pilot.
Okay, I'm going to say a simple thing and then see if you can give me a similar simple thing related to this loss of immune capability. I've been told that this bone density problem, the real mechanism is in our bone marrow. We stop producing these bone cells. Is that accurate, first of all?
In the bone, we're seeing the pre-osteoblast number go down. So it's more than likely due to a lack of proliferation. Okay. In your body right now, you have new bone cells being made, and you have an osteoclast that's remodeling.
Right.
And in spaceflight, it seems like the race between the builder and the destructor is out of kilter.
Is out of kilter, okay. Can you give me a simple type of explanation like that on what's going on in the trigger that's really causing this immune system to back off a little bit and only be about 80% or so effective?
That's exactly what my lab is looking at right now. At night, I go in and do email. We're trying to piece it together, the power of a gene array that looks at 30,000 different genes allows us to start going backward in those pathways that cause the immune cell to be turned on. And we hope to have some really definite answers in the very near future.
So is it fair to say then that the immune cells just aren't being turned on at the same rate that they are in 1G, but the cause of that still needs a lot of work?
It needs work, but we're getting closer.
Okay.
I think within the next 10 years we'll have those answers, and I think those answers will also help people on Earth that are immunosuppressed. So
thank you.
There's a lot of hope.
Thanks for your excellent research.
Thank you.
I have one more question on the bone thing. My mother last year died at the age of 101, and the last couple of years she would fall. She never broke anything. One time she landed on her wrist. She got 2 little tiny scratches and got a multi-drug staph resistant bug. She fell many times. Nothing broke at 101. So you were looking at these different genes, I presume, to try to figure out. And how much progress have you made in finding or in teasing any of that out?
The only problem is getting funding to look at it and getting permission from the astronaut corps to look at it. It's only 2 problems: money and permission.
Okay. Well, what about the non-astronaut corps? The people that are very good or very bad in that aspect.
You could do that, but I don't know of a lot of studies that are dedicated to it, mainly because doing an entire genotype of a person right now is running about $1,000. Okay. And with the NIH funding at the level it's at, it's very hard to get money to do genotype testing because they call that fishing. It's a fishing expedition, whereas really it's just a fact-finding expedition. So it's money, like everything in the world.
Thank you.
Got one more.
I wondered if label unstable cells, type cells, also sensitive for the gravities.
If
label and stable cells.
Cells that are resting.
Right.
And getting them to grow. They still have trouble being activated. Usually when I do an experiment, I put cells into orbit that are at resting, and then I activate them. And I do that so I don't have any conflicting problems with launch vibration and that type of thing. So they still have trouble in being activated. And that's true of the immune cells and the bone cells I've looked at.
What about the skin?
Skin?
Yeah.
Skin infections were seen, I believe, on Skylab, and I think around 2% of all the shuttle people for a 10-year period, there's some skin infections, but I don't know anything more about skin replication.
Thank you so much.
Thank you very much, Millie. Let's give her a round of applause here.